afternoon. Once more, raise your hand if you're going to meet us for the field trip on Sunday.
I'm planning to.
Awesome. It's going to be fun. I want to be sure I have enough forms and field guides. And again, and if you're raising a hand, is it just you or are there multiple people coming with you? Raise two hands.
You mean like family or friends?
Family or friends.
I haven't consulted with them yet.
Okay. Okay, like this. Okay, good. We'll be fine. So meet us at the little school bus at Bachman Pullout off of University by 1:15 on Sunday, because we're going to drive out of there at noon. Don't be late or we'll leave you behind.
Where's that at?
Bachman Pullout is by this bus stop, right off of University and Dole Street.
Oh, just write it down.
Okay. This is a great day. Dr. Ken Kaneshiro is an eminent evolutionary biologist. He received his undergrad in zoology and advanced degrees in entomology here at University of Hawaii. He's currently a researcher at Pacific Biosciences Research Center and program director for the Center for Conservation Research and Training. He's been a key player in developing an understanding of the dynamics of sexual selection and the role it plays in the speciation process, a process we now understand through what's become known as the Kaneshiro hypothesis. We are very fortunate to have him with us today to tell us about it himself. Join me in welcoming Dr. Kaneshiro.
Am I on?
Yes.
Okay, thank you.
I'll go this way. Turn the lights off.
Just the front.
You do it that way.
Okay, I don't know how many years I've done this, but anyway, it's a pleasure to return to talk about, actually I'm not really going to talk about my hypothesis very much. I'm going to mention it at the end, but the what I've been asked to talk about is sort of the tremendous fauna of insects in the Hawaiian Islands. And I'm going to focus on the group of insects that I've been working on for 50 plus years. Just give you a flavor of the kinds of research that we've been doing. As I said, for 50 years, and we've had 75 or 80 senior scientists from all over the world, 400 or 500 students that participate in our project, and there's no way that in this 1 hour period that I'm going to be able to tell you about the results of the research that we've been doing. I think I'm going in and out, yeah.
You're fine.
Okay. Unfortunately, the technology somehow got changed, and so I'm not going to be able to talk like I usually stand in the middle there and use the remote control to advance the slides, but I'm going to have to stand back here and reach over and advance. So bear with me on this one. So I usually start off my talk with this particular slide, and the title, "Nothing in Biology Makes Sense Except in the Light of Evolution." And this was the title of a talk that was presented by Theodosius Dobzhansky in 1972 before the National Association of Biology Teachers. And the reason why I use this is because Hawaii really is the best place in the world to study evolutionary processes. And it really gives us a chance to not only test some of the classical ideas, classical concepts of evolutionary biology, but also to formulate new ideas. And as Cindy mentioned, I was fortunate enough to be working on a group of insects, which I'll talk in the second half. Wait a minute, is that no, I'm talking about that in the first half. About the Hawaiian Drosophila, which led me to develop this idea, this hypothesis on the dynamics of sexual selection in the speciation process. As I said, I'm not going to have time to really get into that very much, but just to mention that Hawaii really is the best place in the world to do these kinds of studies. Now, Cindy, is this sort of the I'm middle of the class right now, the course. So much of the kinds of things that I'm going to be talking about in this first couple of minutes should be reviewed to you folks. So, so the whole thing is what makes Hawaii so special for understanding why is it the best place in the world to do evolutionary biology research. And these are some points that I've just put on the board on the screen here. And I'll talk a little bit about each one of these, but not spend too much time because I want to get into the meat of what I'm here for. So isolation, sequential formation of islands. We know the ages of these islands. There's also fragmentation within islands, not only valleys, but these kipukas that I'm sure you've already heard about. And we have this tremendous diversity of habitats from very dry, desert-like conditions to wet rainforest situations. And then this tremendous diversity of plants and animals that we find in the Hawaiian Islands. And then there are these very special groups of insects, like the Hawaiian Drosophila that I'm going to talk about, which enable us to do very sophisticated research on all aspects of biology, all the way from simple morphology and systematics to analysis of DNA and that sort of thing. So Hawaii, as we all know by now, is the most isolated group of islands in the world, right? The closest landmass is 2,000 miles or so all around the islands. So we don't have a pointer, do we, Cindy?
No, I don't.
Yeah, sorry, I should have brought one. So, the islands are really sitting in the middle of the Pacific Basin, isolated from closest landmass. Oh, thanks. Great, thank you. Yeah, great. And so, and the other thing is about the known ages. So we have pretty good geological data about the ages of these islands. And of course, you got to remember, there's all of these Northwest Hawaiian Islands, all the way from the Emperor Seamounts to Curie, not Curie, but Curie, Midway, and so on, until we have the present High Islands. And Kauai is approximately 5 million years old. And in sequence, we have Oahu, the next oldest, Molokai, Maui, and Lanai, and then Hawaii Island is the youngest island, about 500, 600,000 years old. And again, as I said, the chronological sequence of these island formation is a very important part of understanding the evolutionary processes that have occurred in the native fauna and flora. So, and also, as I had on that initial list, this tremendous diversity of habitats, all the way from very dry conditions, and a lot of it is dictated by the prevailing trade winds, which blow about. Well, of course, now with El Nino and these other global climate issues, much of that has changed, but for the most part, we have trade winds blowing 75% of the time on the windward side of the island, and so you have the wet, rainforest type conditions on the windward side, and by the time you get to the leeward side, it's sort of a dry, more desert-like condition. And you can see this very dramatically on places like the Big Island, where you could have an area like this, which has about 25 inches of rainfall a year on the leeward side. And then on the windward side, in the Waikomoi Forest Reserve, for example, something like 250 inches of rainfall a year. So very extreme climatic conditions. And on the island of Kauai, in Mount Waialeale, which is considered to be the wettest spot on Earth, 500 or 600 inches of rainfall per year. And then, as I mentioned, you have not only separate islands, but also islands within islands by this active volcanism creating these islands of vegetation. So this light spot here is actually a continuous forest with this one here, but with recent lava flows, it's now separated this kipuka from this one here, and that's only about 100 years old. But yet we're finding we could find this, essentially the same species of our Drosophila in a pupaca like this as we find here. But even within 100 years, which is a very short evolutionary time, we're finding significant gene frequency difference, not fixed differences so that they're, would be considered to be different species, but significant gene frequency differences, which is telling us that there's already evolution toward differentiation in species. So the fragmentation, of a forest like this into these separate kipukas are an important part of the evolutionary process in the Hawaiian fauna and flora. And there's, considering the size of the Hawaiian island, which is like, what is it, 0.2% of the total land mass of the United States, or you can stick all of the land mass of the Hawaiian Islands into the state of Connecticut, pretty much. That size, and also the young age of the main Hawaiian Islands, we have many more unique endemic species in the Hawaiian Islands than any place else in the world. So this is this is why Hawaii, the Hawaiian Islands, is a terrific place to conduct these kinds of, better than the Galapagos, that where Darwin himself formulated his ideas and his theory of evolution. Hawaii is actually better than the Galapagos. So you have all of these plants, and I'm sure if you haven't, there hasn't, I don't know if somebody's already talked about plants in Hawaii, the unique native avifauna, the Hawaiian land snails, and then you have the insects. So I guess that's my job today, is to convince you that we have a native Hawaiian insect fauna that's just spectacular. At the present time, we have about four, maybe close now to 5,000 species of insects that are found only in the Hawaiian Islands. Endemic to the Hawaiian Islands, found no place else in the world. However, in the collections, both at the Bishop Museum and our collection here at the University of Hawaii, we know that there's going to be close to 10,000 species of insects unique to the Hawaiian Islands. So taxonomically, systematically, we're only halfway there in naming and describing all of the species that we know will exist in the Hawaiian Islands. And then, as I mentioned, there are these very special groups of insects and plants and other organisms in Hawaii that enable us to do very sophisticated research in the laboratory, in the field, to better understand evolutionary processes that gave rise to this tremendous number of organisms. So I'm going to spend the next few minutes talking about as I said, we've had more than 50 years of work, more than 500 scientific papers already published on this group. We started in 1963 as a joint project funded by the National Institute of Health of all federal agencies. It started in 1963 as a joint effort between the University of Texas at Austin and the University of Hawaii. And you ask why University of Texas at Austin? At that time, University of Texas had what was called the Genetics Foundation, which was funded by both the National Institute of Health and the National Science Foundation. And it was the director of the Genetics Foundation at UT that saw the potential of studying Hawaiian Drosophila to understand better evolutionary processes. So he put together a team and got them in a partnership with the University of Hawaii, primarily with Professor Elmo Hardy, who was a professor in the Department of Entomology. He was my major professor. And together they put together a team of researchers, not only in systematics and understanding the taxonomy of this group, but insect ecologists, behaviorists, geneticists, a whole team of multidisciplinary scientists to understand this group. So 1963, that was 50 plus years. I started on this project in June of 1963 as a sophomore here at the University of Hawaii. And I took a job as a dishwasher for the project, washing the vials and the test tubes and so on that we use to culture these flies. And I stayed with it and continued to study this group. As I said, it's a multidisciplinary team that came and looked at this group in a systems approach. So I already mentioned this. We have more than 500 species already named and described, but there's already another 250 or 300 more species that we know are new and undescribed that's already in the collection, both, well, not so much in the Bishop Museum, but most of the collection here in the University of Hawaii. And so, you know, and we still, even after 50 plus years and even going back to some of the classical sites, we're still discovering new species. And so my conservative estimate is that there could be as many as 1,000 species just in this one family of insects in the Hawaiian Islands. And the most amazing thing is that potentially 1,000 species probably arose from a single introduction that made it across 2,000 plus miles X million years ago, that made it to the Hawaiian Islands and then evolved to this tremendous number of species. So this is just a collage of a series of shots of some of the Hawaiian Drosophila. How many of you know about or have heard about Drosophila melanogaster in your basic biology class? Okay. So you know what they look like, right? They're pretty tiny, nondescript yellow-bodied flies. But our Hawaiian Drosophila have these. It's actually the giants, the largest Drosophila, largest species in the family Drosophilidae are from Hawaii. It's just, as I said, there's no way I'm going to be able to show you the 500 or potentially 1,000 species, but it just gives you a flavor of the spectacular group of insects that we have in Hawaii. The other thing about Hawaiian Drosophila, you know, the Drosophila melanogaster, most genetics laboratory elsewhere in the world call them fruit flies. Okay, and in Hawaii, we've got to be very careful because Hawaiian Drosophila, if you call them fruit flies, we also have the true fruit flies in a totally different family, right? The family Tephrididae. And the true fruit flies are agricultural pests. They lay their eggs in the papayas, your bananas, and your fruits, your guavas, and so on. And there is an effort to control fruit flies, true fruit flies, to protect and to even enhance the agribusiness, the agricultural industry. So we want to be sure we don't confuse fruit flies, which is what genetics laboratory elsewhere in the world called Drosophila melanogaster and the other species that they study. But in Hawaii, we've got to be very careful because we do have the Mediterranean fruit fly, the Oriental fruit fly, melon fly, which are all important agricultural pests. And the USDA and others in Hawaii are making an effort to control these fruit fly species. Okay. The Hawaiian Drosophila are, some of them do use fruit, but for the most part, they, in the Hawaiian, native Hawaiian ecosystem, there are very few plants that have fruits that are very large enough. They're not like a papaya or a mango or a guava. Most of the fruits are pretty tiny. And so in order for the large Drosophila that we have in the Hawaiian, native Hawaiian fauna, the tiny fruits are not enough of a resource for the development of the larval stage. So what happens is that they've used almost everything else in the native ecosystem, the rainforest and so on. So ferns are used, decaying parts of the ferns are used. This particular plant does have a fruit that's relatively large. It's in the genus Claremontia, or in the group Lobiliaceae, which is fairly large. But and there are some Drosophila species that use that as a larval breeding site. But they also use the decaying leaves, the decaying branches, and even the roots sometimes of this particular group are used as larval breeding site. Leaves, tremendous biomass of leaves that fall in the native rainforest. And almost every species of native plant, the leaves, have a Drosophila species in the leaf breeder group use the decaying leaves. Fungi, another important larval resource. So there's a whole group of fungus-feeding Hawaiian Drosophila. This particular fungus, it's a shell fungus, is about 3 feet across in diameter and about 4 feet tall. So it's a huge piece of fungus that we found on the Big Island on one of the ohia trees that was growing there. And I mean, again, this is just to show you that there are these potentially huge biomass of larval resource that our Hawaiian Drosophila use as a breeding site. We have some flower breeders. So morning glory flowers. Morning glory flowers, however, are very ephemeral. You know, they're sort of in a vegetative state for most. The plant is in a vegetative state for most of the time, and then the flower blooms and dies within about 24, 48 hours at the most. So in that time, these are our Hawaiian Drosophila that's in the flower head. They actually will lay their eggs, and the eggs will hatch within that day, and the larvae will begin to feed on the decaying flower head. So flower breeders. Here's another silver sword plant, in which the flower heads are also used as a larval breeding site for one of our Hawaiian Drosophila species. And I don't know if somebody's already talked about silver swords, but this particular plant, which is in, this one is in Haleakala crater, it's in this vegetative, so these are sort of the leaves of the plant, and they're in that state for about sometimes 10 or 15 years. And then it puts out this flower head, composite flower head. And that's the only time that our Hawaiian Drosophila are found on this plant, feeding and laying eggs, and larval develop in the flower heads. The problem is there are periods in Haleakala crater in which all of the silver sword are in this vegetative state, and they could be periods of a couple of years or more in which there are no silver sword with this flowers. Where are the flies in those 2 years? Because the adults obviously can't live for 2 or 3 years. Are the eggs or the larvae and or the pupa of the Drosophila in some kind of a diapause or what? Where are they in the 2 or 3 years when there are no silver sword plants with no flowers? We don't know. It's still a mystery to us. So here's a great project for a graduate student, although they may have to work for 2 or 3 or 4 years to really figure this out, because it's when there are no plants with flowers during the 2 or 3 years period that we need to do more investigation. I mean, that's a long time for a master's or a PhD student, and so that's a mystery to us. There are some species that have become adapted to feeding or being a predator on a native spider, the egg mass of a spider. So this is an egg mass of a spider, and right here in this corner is an egg of one of our Hawaiian Drosophila. Here's a larva that has fed on the spider eggs, and here's a pupa. So it spends its entire life cycle on the underside of the leaf where the spider eggs are found. And so here's another weird adaptive shift from, Drosophila from elsewhere in the world mostly feed on fruits. That's why they're called fruit flies. But here's a shift to becoming a predator on spider eggs. Here's another very weird adaptation. We discovered, this was a trip but this is actually me when I was an undergraduate student. And we were actually in the Kohala Mountains, and you go all the way to the end of the road, you rent some mules, because we have to hike in over 3 valleys to get to the site where we're interested in. And as we were down at the bottom of the valley and crossing the stream before we get up to the next valley, as we were crossing the stream, we found on these boulders some flies. As the mule passed, we saw some flies fly out. And we thought, oh, they're just typical aquatic flies, flies that you would normally find in the middle of the stream. Got down on our hands and knees, and we collected some of these flies, and we looked at it with our hand lens, and it turned out to be a Drosophila-like species. couldn't believe that the Drosophila would be sitting in the middle of the stream, but then we started to poke around and found in places like this, there's some green algae below the surface of the water. And if you were to scrape it up with your fingernails, there were some larvae there. Again, take our hand lens, look at the larvae. Turned out to be the larvae of a Drosophila. Now, we weren't prepared to make this discovery, right? It was just accidentally that we were crossing the stream, we saw these flies. Turned out to be a Drosophila. Couldn't believe it, but we collected several bags of water, stream water, the algae and the larvae. We brought it back to the laboratory here, so this is on a big eye, so we flew back to Honolulu, put it, got it back to our lab, but we found that all of the larvae had suffocated and died, right? Because, unless you're aerating the water, they don't have enough oxygen to survive. So we were never able to confirm that the larvae that we found below the surface of the water in the algae were actually the